Welcome to the deep dive. Today we're getting into the core of a really fundamental shift that's happening in modern biology.
It really is.
For years, so much of the work was observational, right? Just watching cells, cataloging what they do. Now we're really moving into a world of active intervention.
Of asking cells very specific questions. And that's the revolution. We're talking about these huge, high-throughput experiments, things like Perturb-seq, CROP-seq.
Right.
Researchers aren't just looking at one thing at a time. They're hitting, thousands of cells at once with different genetic tweaks, different drugs or disease states, and then capturing the response of every single cell.
And when you start doing that to millions of individual cells with thousands of different stimuli, you get these absolutely gigantic, complex data sets.
Sure.
The challenge is completely flipped. It's not about generating the data anymore. It's about figuring out what on earth to do with it.
Precisely. And the problem is a lot of the established analysis tools are, they're siloed. They might be stuck in the older R ecosystem. Or maybe they were built for just one very specific kind of CRISPR screen.
And they just fall over when you show them genome scale data.
They just collapse. And critically, they often analyze the raw numbers in a vacuum. They don't integrate all the biological knowledge we already have, the metadata, the context about what these genes and drugs actually do.
Which brings us to the solution we're discussing today, Pertpy.
Exactly. Pertpy is designed to bridge that exact gap. It's an end-to-end modular framework built in Python, and it's aimed squarely at organizing, analyzing, and visualizing these massive experiments.
It sounds like a pretty big promise.
Yeah.
What makes Pertpy scalable where other tools have failed?
I'd say two things are absolutely key. First, its foundation. Pertpy is built to be part of the scverse ecosystem.
Okay, so if you're in single cell and using Python, Scampi and tools like that.
Right. Pertpy is designed for seamless interoperability with them. It breaks down those technical silos. But second, and this is huge, is speed. Pertpy uses the high-performance computation library, JAX.
JAX is so important. It's famous in machine learning circles. What's it doing for biology?
It just brings substantial performance gains. JAX allows for sparse, memory-efficient math, and it uses GPUs to the absolute fullest. So for you, the listener, this means Pertpy's versions of old methods are often way, way faster than the original ones.
And that speed is what you're going to need for the next generation of projects.
It's an absolute requirement when you start talking about hundreds of millions of cells.
Okay, let's unpack this pipeline, starting right at the beginning. You do a CRISPR screen. The first massive challenge is just figuring out which genetic modification went into which cell. We need to talk about guide RNA assignment.
This is step one, linking the cause to the effect. Pertpy gives you a flexible toolbox here. If your experiment is clean, low multiplicity of infection, meaning most cells got just one guide, you can use a simple thresholding model.
So it just assigns the guide that's most expressed, as long as it's over a certain count.
Exactly. Simple and effective for clean data.
But single-cell data is never clean, right? It's famously noisy. What if things were more ambiguous?
That's where the heavy-duty stats come in. Pertpy has a more sophisticated tool, the Poisson-Gaussian mixture model. This doesn't just use a fixed threshold. Instead, it statistically classifies cells based on whether they belong to a high-expressing population for a given guide.
So it's learning the difference between background noise and a real signal.
Precisely. And that makes it incredibly robust for both low and high MOI screens. It's just a more nuanced, statistically solid way to make the call.
Okay, so we've assigned our perturbations. Now we hit the next big wall. Confounding factors, batch effects.
The enemy of all large studies.
Right. If one plate was processed on Tuesday and another on Friday, that technical noise can just drown the real biology, especially for subtle effects.
You're at risk of finding a false discovery, mistaking an artifact for a real interaction. Pertpy's solution here is, it's elegant. Because it's in the scverse, users can just plug and play with established batch correction methods that are already in Python.
So it doesn't have to reinvent the wheel.
It just integrates the best tools that already exist.
Now we get to the really exciting part, turning raw numbers into biological gold, integrating context. How does Pertpy actually enrich this data?
This is one of its most powerful features. It moves beyond just gene counts by automatically pulling in metadata from established biological databases. We're talking about the cell line ontology, the drug ontology.
So if my data is from, say, K562 cells, Pertpy just knows what that is, its origin, its properties.
Not just that, it enriches it further. For cell lines, it pulls from the cancer dependency map, depth map, and GDSC. For drugs, it grabs mechanism data from the connectivity map, CMAP, and chemical details from PubChem and Chembol.
That context is what turns a list of genes into a discovery about a pathway.
Absolutely vital. And there's one last critical quality control step before we even get to the analysis.
Checking if the perturbation actually worked. The escapees.
Exactly. Just because you hit a cell with a CRISPR guide doesn't mean the gene was actually knocked out. They are a huge source of noise that can completely dilute your signal.
So you average in a bunch of cells where nothing happened.
Yeah.
And your effect looks weaker than it is.
Or it disappears completely. So Pertpy includes a fast, JX-optimized implementation of a tool called Mixscape.
And how does Mixscape computationally separate the successes from the failures?
It's ingenious, really. For every single cell that was targeted, Mixscape finds its closest neighbors in the data that were control cells.
Okay.
It then compares the expression signature of your targeted cell to that local control neighborhood. If the targeted cell's signature is way different, it gets labeled KO knockout. It worked.
And if it looks just like its control neighbors,
it's labeled NP not perturbed. An escapee. It failed.
So Mixscape is basically a computational guarantee that your downstream analysis is looking at actual biology, not just experimental noise.
Yes. That single step dramatically increases your confidence in any findings. It gives you that clean, robust, contextualized foundation you need.
Okay, that was a lot. Let's just take a moment to recap that foundational pipeline.
Good idea.
So first, Pertpy gives you a structured pipeline that starts with robust gRNA assignment to link the perturbation to the cell.
And critically, our first key takeaway is that it offers two models for this. A simple threshold for clean data and a more advanced mixture model for noisy or complex high MOI screens.
Then, it leverages the whole scverse ecosystem to handle technical artifacts like batch effects. And then, takeaway number 2, it enriches that raw data with crucial biological context from external databases like DepMap, CMAP, and PubChem.
And our third key takeaway is that it performs essential quality control with Mixscape, which computationally identifies and separates the truly perturbed cells from the escapees, ensuring your analysis is based on a clean signal.
Excellent. So with that clean foundation, we can move into the deep analytical modules. And this is where there's a really important conceptual leap. We move away from looking at thousands of individual cells to generating a single, concise summary of the collective impact of a stimulus. We're creating a perturbation space.
Think of it like this. Instead of trying to look at 5,000 unique photos of cells that responded to drug X, we're going to synthesize them all into one representative avatar of what drug X does.
I like that. An avatar.
And that avatar becomes a single point in this new perturbation space. And so perturbations that do similar things to cells will naturally cluster together in this space.
So how do you calculate that avatar? What's the method?
Well, one really powerful approach is the multilayer perceptron MLP classifier space. Pertpy trains a neural network to predict which perturbation was applied to each cell.
Okay, so it learns to tell the difference between drug X and drug Y.
Exactly, but here's the clever part. We don't actually use the network's final prediction. Instead, we extract the representation from the last hidden layer before the prediction.
So you're grabbing the summary of features the network decided were most important for telling the perturbations apart.
That's it. That learned feature set becomes your embedding, your avatar. You can also use simpler methods like a pseudo-bulk space, which is basically just averaging gene expression. But the MLP space gives you this really functionally relevant grouping.
Okay, once we've mapped our perturbations, we have to dig into the classic analysis. Differential gene expression, finding which genes actually change.
DGE is fundamental, but standard tools can really fall short when your experiment gets complicated. Multiple conditions, multiple batches, nested comparisons.
You want to compare drug A in one cell line versus drug B in another, while controlling for the sequencing run.
Right, and that gets messy. Pertpy gives you a unified interface to handle that complexity. It supports advanced statistical frameworks like PyDissect2 and Edger right alongside classic t-tests, all in one place.
So it's about handling modern experimental design without juggling a dozen different packages.
Precisely. And that complexity extends beyond genes to whole cell populations. If you treat a tumor sample, some cell types might grow, others might shrink. You need to detect these compositional shifts.
And the sources mentioned Pertpy handles 2 different scenarios for this, depending on your data.
Correct. The first scenario is when you have distinct labeled groups, your T cells, your B cells, etc. For this, Pertpy offers these really fast Bayesian implementations of Secoda 2.0 and Tascoda 2.0.
Why Bayesian? What's the advantage there?
Because they correctly model the sum-to-one problem of compositional data. You know, the fact that if one cell type increases, others must, by definition, decrease as a percentage. Bayesian models handle that uncertainty very robustly.
Okay, that makes sense for clear groups. But what about scenario 2? When cells aren't labeled, or it's a continuous process like development, where the boundaries are all fuzzy?
For that, Pertpy has a scalable implementation of a tool called Milo. Now, Milo works by building a k-nearest neighbor graph across all your cells. Then it looks for changes in cell density across overlapping neighborhoods on that graph.
So you don't need sharp boundaries, you just look for regions in the graph that get more or less crowded after a treatment.
Exactly. It's a high-resolution method that's perfect for continuous or poorly defined systems.
That flexibility is powerful. Okay, finally, how do we measure the impact? How do we quantify the difference between drug A's effect and drug B's?
We need robust distance metrics, and Pertpy implements over 18 of them, because no single metric is right for every biological question. You have to pick one that fits your assumptions about the data.
Okay, don't list all 18, but give us a sense of the range, the simplest versus something more advanced.
Sure. On the simple end, you have things like mean squared error, MSE. It's basically the average squared difference between the two responses. It's fast, but it can be sensitive to outliers.
And on the advanced end?
You have optimal transport-based distances, like the famous Wasserstein distance.
The Earth mover's distance.
Yes. You can think of it as what's the minimum cost or work required to transform the distribution of cells from perturbation A into the distribution of cells from perturbation B. It's much more sensitive to changes in the shape and density of the data.
And I assume you can test if these distances are meaningful.
Oh, absolutely. Every single one of these metrics can be tested for statistical significance using Monte Carlo permutation testing. So you can be confident that the difference you're measuring is real.
That's a huge amount of analytical firepower. Let's do another quick recap before we get into the case studies.
Sounds good.
So we've moved from data prep to analysis. A huge concept here is takeaway number 4, the perturbation space, which summarizes the collective response of thousands of cells into a single representative point, or avatar, letting us see how different stimuli relate to each other.
And for analyzing cell abundance, takeaway number 5 is that Pertpy is flexible. For labeled data, it uses Bayesian tools like Sakota, and for unlabeled or continuous data, it uses the graph-based tool Milo.
Finally, takeaway number 6. To quantify the effects, Pertpy offers over 18 different distance metrics, from simple MSE to complex ones like Wasserstein distance, and all of them can be rigorously tested for statistical significance.
That's a great summary.
Okay, this brings us to the most compelling part. What did this framework actually uncover? Let's talk real-world biological insights.
Let's start with case study 1, finding new gene programs in a big CRISPR activation screen. This was the Norman et al. Study, over 111,000 cells. Pertpy used that MLP classifier space we talked about to refine the original findings.
So what was the key insight?
Well, it confirmed a lot of known biology, but the clustering revealed deeper truths. For example, perturbing the gene TP73, which was thought of as a pioneer factor, unexpectedly clustered right next to strong G1 cell cycle perturbations.
So the main effect, the avatar of TP73 activation, is a cell cycle effect.
Exactly. It refines our functional understanding of that gene based purely on the cell's response. It's like using the data to update the textbook. And it went further. Pertpy actually split a big, old cluster called pro-growth into 2 distinct subgroups.
What were they?
One was focused on cripple-like factors, or KLFs, and the other was focused on MAPK encoding genes. So it revealed 2 completely distinct biological mechanisms for driving growth that had previously just been lumped together.
That's impressive. Okay, case study 2. Deconvolving drug response. This tackles a really tricky problem, separating the effect of a drug that just kills cells from the more subtle changes that happen before death.
Yes, this was the MXSEC dataset across 172 cell lines, and it's a beautiful example of integrating that external metadata we talked about. Pertpy first goes and fetches the drug sensitivity scores for each cell line from a database like GDSC.
Okay, so it knows which cell lines are tough and which are sensitive to a given drug.
Right. Then it uses a really neat statistical model. For every single gene, it fits a linear regression.
And how does that simple regression separate out the viability effect?
You model the gene's expression change as a function of the cell line's sensitivity. The model gives you 2 key values, an intercept and a slope. The intercept is the viability-independent response. It's the change that happens in every cell line, regardless of whether it lives or dies.
That's the core baseline effect of the drug itself.
Exactly, whereas the slope is the viability-dependent response. These are the gene changes that are directly tied to how sensitive the cell line is to being killed by the drug.
So what new discovery did this reveal for the BRAF inhibitor, dibrafenib?
It revealed two totally different stories. The viability-dependent response, the slope, was enriched for interferon signaling genes. That's what you'd expect for immune-mediated cell death. But the viability-independent response, the intercept, what's happening even in resistant cells, showed the surprising upregulation of protein translation pathway genes.
A completely new mechanism separate from BRAF inhibition that you'd never have seen otherwise.
It would have been completely buried if you just averaged everything together.
Incredible. And that leads us to the final case study, which has direct clinical relevance in triple-negative breast cancer, or TNBC.
Right. Here they were looking at patient response to chemotherapy, with or without a new immunotherapy. The focus was on the tumor microenvironment.
Tracking immune cell shifts.
Yes. Using SCODA, Pertpy looked for changes in the composition of immune cells, like CD4 and CD8 T cells, before and after treatment. And they found significant shifts only in patients who got chemotherapy alone.
But not in the combo therapy group.
Not in the combo group. It suggests the combination therapy might be modulating the T cell environment in a fundamentally different, maybe less drastic way, which could have implications for prognosis.
And they went even deeper, looking at how cells talk to each other.
They did, using Pertpy's implementation of a tool called Dialogue. They found these multicellular programs, or MCPs, which are like functional units of interacting cells in the tissue. One of them, MCP2, seemed to be associated with a good response to treatment.
And what defined that program?
It was linked to an interaction between the cytokine IL-7 and its receptor on T cells, which is important for T cell survival. And crucially, they saw an increase in genes like JUN, FOS, and FOSB. These are key parts of the AP1 complex, which is known to affect T cell activation and, potentially, their anti-tumor activity.
So Pertpy is really the full toolbox, from the basic gRNA assignment all the way up to these complex intercellular communication metrics that can generate clinically relevant hypotheses.
That's the big picture. It takes all these powerful tools, many of which were stuck in R or were too slow, and brings them into this fast, interoperable Python and scverse ecosystem. And it's all built with future scale in mind.
Yeah. And that scalability is the final frontier, isn't it?
Yeah.
We're already seeing data sets that are aiming for hundreds of millions of cells.
Absolutely. The speed you get from JAX today and from things like Dask integration tomorrow is going to stop being a nice-to-have and become a non-negotiable requirement.
This is what will enable the creation of true, large-scale perturbation atlases.
Comprehensive maps. Maps of how every cell type we know reacts to every possible condition, moving way beyond just cataloging cells in their resting state.
So here is our final provocative thought for you to take away from this deep dive.
Here.
If a framework like Pertpy allows researchers to build these massive perturbation atlases and then train foundation models AI trained on a map of every inducible cellular state, what profound, non-obvious impact do you think that will have on personalized medicine and drug discovery in the next 5 years?
It feels like the knowledge we're synthesizing today is really just the starting line for a pretty massive revolution in how we understand and engineer biology.